Hello, I'm Elad Sharon from the Hebrew University in Jerusalem, and in this work I'm going to present a method to find new photosynthesis-related genes using coevolution analysis. It cannot be overestimated that photosynthesis is one of the main processes on Earth. In this process, plants and other photosynthetic organisms turn solar energy, water, and carbon dioxide into sugars and oxygen through the light and dark reactions. Finding new genes in such an important process can have high impact by improvement of photosynthesis process, resulting in increased biomass and eventually, in agriculture point of view, increase in yield. In this study, we use phylogenetic profiling. In this method, we assume that proteins that function in the same pathways, in the same complexes, or the same environmental conditions are likely to show similar presence or absence pattern across phylogenetic clades. To generate phylogenetic profiles, we collected extensive information of many proteomes integrated from UniProt, Ensemble, RefSeq, and NR database to create a unified database that represents different clades. We then blast Arabidopsis thaliana proteome against this unified database and generated co-evolved clusters using hierarchical clustering based on Pearson correlation. Next, we wanted to find the best representative cluster of photosynthesis, so we used the F1 measure, which is the harmonic mean between precision and recall. So taking as a query the Go term of photosynthesis, we calculated the F1 measure to each co-evolved cluster and choose the cluster with the highest score to be the representative cluster of photosynthesis. Since the coevolution score is based on BLAST, there can be false positive results derived from genes with similar sequences. So we grouped together genes with similarity in their sequence and referred to each group as a single gene in our calculations. The cluster we found consists of known annotated and unannotated photosynthesis-related genes. The heat map shows the genes in the rows and the organisms in the columns. As the heat map shows, the genes are mainly occur in photosynthetic organisms like plants, green algae, brown and red algae, diatoms, and other known photosynthetic protists. So this pattern that shows occurrence only or mainly in photosynthetic organisms look promise to classify these clusters as photosynthesis-related cluster. Moreover, to evaluate that this cluster is unique for photosynthesis and nothing else, we conducted Go enrichment analysis and found that the cluster is enriched by photosynthetic-related pathways like light harvesting in photosystem 1, photosystem 2 assembly, photosystem 2 stabilization, and more. In numbers, the resultant cluster consists of 146 genes, of which 46 genes shown in green are from the query, 70 genes are known to be involved in photosynthesis-related pathways, and approximately 40 genes have no annotations and can be potentially new photosynthesis-related genes. We further examined the interaction in the cluster. So we analyzed the 70 genes involved in photosynthesis-related pathways using string. The network we got showed that most of the photosynthesis-related genes in the cluster have been identified in co-expression and interaction analysis. But there are some genes that don't show co-expression or direct interaction and were connected by the coevolution analysis. So in conclusion, phylogenetic profiling analysis identify a unique cluster of photosynthesis-related genes. The photosynthetic cluster contains photosynthesis-related genes that weren't identified in co-expression and interaction analysis, emphasizing the power of phylogenetic profiling to predict genes function. So we think that phylogenetic profiling can be applied to economically important crop plants to find new genes that related to photosynthesis, to improve their photosynthesis, and eventually help to increase yield in those plants. Thank you very much for listening.